All right. Hello, everyone. My name is Chamteut Oh. Thank you for giving me the chance to share my research findings with you. I'm an environmental engineer. I'm from University of, I'm PhD candidate, I'm sorry. University of Illinois, Urbana-Champaign. And I'm an incoming assistant professor in the Department of Environmental Engineering Sciences at the University of Florida, starting fourth semester. Many research presentations at this conference already proved the effectiveness of Far-UVC in pathogen inactivation. In our study, we also confirmed effectiveness of Far-UVC in adenovirus. But I'd like to focus more on understanding the mechanisms of microplasma UV lamp for adenovirus inactivation. So I hope this presentation provides you with insights into why Far-UVC is very effective for virus inactivation. I'd like to first express my acknowledgement. This study was funded by US EPA, and microplasma UV lamps were provided by Eden Park Illumination. I'm also grateful to have worked with many experts. Professor Wien, who is my PhD advisor, served as the PI for this project. And Dr. Sun is the expert in microplasma UV lamp, and Dr. Araut specializes in virology. So this presentation is all about the application of the emerging UV technology, which is Far-UVC, for water disinfection. UV irradiation has played a huge role in water disinfection because of many advantages. And I'd like to emphasize two specific aspects that are particularly relevant to water disinfection. First, basically, UV irradiation can complement the limitation of chemical disinfectants, such as free chlorine. So unlike free chlorine, UV doesn't produce disinfection byproducts, which are carcinogens. And second, the UV irradiation proves its effectiveness against protozoa, such as Cryptosporidium and Giardia, which are very highly resistant to free chlorine. Low pressure mercury UV lamp has widely used for water disinfection. And as you may know, this lamp generates monochromatic UV wavelength at 254 nanometer, which are known for its ability to damage nucleic acid. But the critical limitation of this lamp is that adenovirus exhibit particularly high resistance to LPUV radiation. This table illustrates the dozens of more UV fluence is required to inactivate adenoviruses compared to the other pathogens. The high resistance of the adenovirus can be explained by its unique ability of the adenoviruses, which is that adenovirus, even though they have a genome damage, they can still penetrate host cells and use host cell machinery to repair their damaged genome and then infect host cells. So genome damage is not lethal to adenoviruses. In this context, the development of microplasma UV lamp caught our attention because of its unique ability to generate 222 nanometer wavelength with high power. Many studies already supports that this specific wavelength could possibly target different virus structure. So the main objective of this study was to investigate the impact of microplasma UV lamp on adenovirus inactivation. To understand the adenovirus inactivation mechanisms, we've developed molecular assays to quantify the integrity of virus structure, such as genome and capsid protein and receptor-binding domain. And by comparing the results from molecular assays to the results from cell culture-based infectivity assays, such as FLAG assay, we can understand how viruses lose their infectivity when they are exposed to disinfection technologies. And through this approach, we revealed inactivation mechanisms of wide range of emerging technologies. And the methodology is described as in these publications. So in the following slides, I will briefly introduce how each molecular assay allows us to understand the integrity of each virus structure. First, we developed an assay that can quantify 10,000 base pair long genome, which accounts for about 30% of complete adenovirus genome. In this assay, we use two types of polymerases. In the first step, long MTAC DNA polymerase synthesized 10,000 base pair long genome. And this long amplicon can be quantified by the conventional qPCR at the second step. So the second amplicon can be produced only when the viral sequence are intact in this long region. Second, we developed an assay that can quantify the number of viruses with lesion on their capsid proteins, through which nuclease can penetrate the virus particles. Nuclease is 5 nanometer long enzyme that can cleave the genome into small pieces. So if the capsid protein is denatured, nuclease can penetrate the virus particle and degrade the genome. Third, we also established assay that can quantify the number of viruses that can bind to host cells. In this assay, we used A549 cell line, which is host cell for adenovirus, to exclude viruses whose receptor binding domains doesn't function well. So we applied 222 nanometer wavelength irradiation to adenovirus inactivation. And this graph shows the result. X-axis shows the amount of UV applied to viruses, and Y-axis shows a log reduction of virus infectivity or integrity of each virus structure. I'd like to first draw your attention to the infectivity loss, which is denoted by black squares. And we can calculate the inactivation rate constant, which is the slope of this graph. And this value was 4.4 times higher than that from the LPUV, generating to 54 nanometer. We also confirmed capsid damage explained the infectivity loss very well, while the other two virus structures, including genome and receptor binding domain, are quite stable under 222 nanometer wavelength. So we concluded 222 nanometer wavelengths primarily destroyed the capsid of adenoviruses, which result in virus inactivation. Please keep in mind that unlike genome damage, protein damage is not repairable. So this damage is lethal to adenoviruses. And this mechanism explains why Far-UVC is highly effective for virus inactivation. So this is all I prepared. Thank you very much for your attention.
Thank you. Are there any questions? Okay, thank you very much.
Thank you.